Cool. Thank you. Thank you so much for the invitation. It's an honor for me to present to you. I apologize I can't be there in person. I'm on clinical service this week. So I'm excited to deliver this talk from the comforts of my office. So today we're going to talk about diagnostics and dilemmas, specifically focusing on parasitological infections. They're becoming increasingly not common, but encountered as we travel. There are a number of patients on biologics who are immunosuppressed, and so they can acquire these infections, remain asymptomatic, and then present subsequently. So I just want to talk about kind of from a lab perspective how we perform diagnostics for a number of these parasitological infections, focusing on a parasite that's near and dear to my heart, leishmaniasis, which is what I did my research on during my PhD, and how it all works, what our current landscape is, and where do we go from here and what's to come in the future. So I have no conflicts of interest. This was my conflict of interest when I gave a similar talk a few months ago, jumping on my lap, but today there's nothing I have to share with you. So quick outline, as I mentioned, we'll go over the history of parasites, parasitology, the diagnostic tools that we currently do have available, dilemmas with some of these diagnostic tools, focusing on leishmaniasis as an example, and what the future does hold for diagnosing these infections in a clinical microbiology laboratory. Okay, so parasites are, as many of you I'm sure are well aware, organisms that live on or in a host organism that obtain food from or at the expense of the host. And there's kind of three main groups of parasites that we think about: the protozoan parasites. So you can see an example on the bottom left of a entamoeba. So these are microscopic one-celled organisms, often free-living and can be found in nature. There are the helminths, which I'm sure many of you are aware of, worms, so the roundworms like nematodes. There's also the flukes, trematodes, and tapeworms, such as the cestodes, and ectoparasites. So these are organisms like ticks, fleas, lice, mites that can be found on a number of animals as well as humans that can spread disease as well. So there's been over 300 different kinds of helminthic worms identified in the world, and about 70 different species of protozoa are known to actually infect humans. And currently, we're dealing with about 90 common species that cause important disease today. And I'm sure many of you are aware of some of them, like malaria, leishmaniasis, strongyloides being some of them. And here I just wanted to show an article from CBC discussing another parasite called schistosomiasis and how they've identified this parasite in a skeleton from more than 6,000 years ago and from an area where schistosomiasis is currently still endemic, so Syria. So we can see not much has changed, and I'll talk about that further with regards to where parasitic infections are endemic and where we can see them popping up. So in terms of the history of parasitology, so just kind of going back and reflecting on the history that's associated with humans. So, Homo sapiens emerged from Eastern Africa about 150,000 years ago, and humans have migrated since then to many parts of the world, bringing in what we call heirloom parasites. So these are parasites that have been inherited from their ancestors, and what we call souvenir parasites, so inherited from other, animals, contact with animals in through the migration process. And ever since then, there's really been a spread and distribution, largely a result of the human activities. And so we can see parasites brought over to the New World, so going in from the Old World to the New World, as you can kind of see in the map that I've depicted on the bottom right. And a good example of this is leishmaniasis, where the parasite was, predominantly found in the Old World setting, and now we have spread towards the New World in the last couple hundred years ago. And, with the advent of AIDS, for example, and patients on biologics and immunosuppression, we see more and more of parasitic infections being encountered because of the opportunistic nature of some of these parasites. And so this I thought was a really neat map that I wanted to share. So it's a global map of not only parasites, but other infections from 1944. And I don't think much has changed with this distribution. And so you can see the red is actually indicative of an areas endemic for malaria. And there's a bunch of other figures that you can see, and all those are representing different kinds of diseases. And so I just wanted to highlight that not much has unfortunately changed with regards to the distribution with some of these diseases and specifically parasites. And so, talking about some top travel-related parasitic infections that are vector-borne, malaria. I'm sure you guys have heard of now kind of found, well, a few cases have been reported in the States, questionable as to whether or not it's established. Leishmaniasis, which I'll talk about further. Contaminated food and water associated parasites like Giardia, Cryptosporidia, and Cyclospora. So these are some, common parasitic infections that we do encounter. And I just wanted to bring up this figure. So this is a table highlighting the 18 groups of infectious diseases caused by, yes, parasites, viruses, and bacteria that make up a group called neglected tropical diseases. And these diseases disproportionately affect those who are often poor, and they cause significant health and financial burden. So these diseases are found in parts of the world where there's poor sanitation. There are 149 countries in which NTDs are endemic. Over 1 billion people are infected, and there's over 2 billion at risk. And these diseases are largely treatable and preventable if we're able to control the vectors that carry these diseases, as well as improving water quality, sanitation, et cetera. So just wanted to highlight the groups with the red star. And so those are the groups that are parasitic in nature and make up a large proportion of the neglected tropical diseases. So talking about parasitology and diagnostic parasitology, how do we identify these organisms in the laboratory? Well, not much has changed since the development of the microscope in the 15th century, but we do have other methods like serology, molecular-based approaches like PCR. I'm sure you guys have heard of rapid diagnostic tests, especially with COVID. There's RDTs for malaria, often used in endemic settings, and other parasites like leishmaniasis. And what's been kind of taking off in the world of parasitology is looking into sequencing and proteomics as methods of diagnosing these parasitological infections. Now, microscopy is the mainstay. It's been used for many years, largely because it's quite easy to perform, but cheap. But it is time-consuming, labor-intensive, and you do need staff who are really well trained in being able to identify some parasitic infections. Some of them look like artifact, and so you want to make sure that you have a really astute technician looking under the microscope to be able to pick up these infections. And microscopy is really easy because it could be done on a variety of samples, on blood, on tissue, stool, lymph nodes, you name it, CSF samples, so cerebral spinal fluid if you are at risk of some CNS infection. So microscopy is really kind of the mainstay in parasitology, and with all these, advancements, we're kind of losing that skilled expertise in microscopy, which is a huge concern, and which is why there's kind of a push for looking into more novel techniques that can be implemented to diagnose these infections, which we'll talk about. But I just want to highlight how important microscopy is. It's kind of the backbone for parasitology in particular. And really a huge diagnostic modality that we do have in the lab across the board. Now there's serological based assays, and so this is typically used, when you can't perform microscopy or direct examination, so to speak. In terms of the turnaround time, like microscopy, it can be quick if you do have the assay available on hand. It is more sensitive and specific, and the highest yield is actually using some of these serological-based assays is when your microscopy is inconclusive. You can't tell if you're looking under the microscope what that organism is, or for example, the patient has very low parasite burden, asymptomatic. There's a number of other factors that can contribute to why the microscopy was unrevealing. So for example, depending on the parasite, there might be low egg production, or the production of eggs is sporadic in nature. You didn't collect the sample at the right time. So really, serology kind of is the backbone for after microscopy for diagnosing some of these infections. Now moving on to the more molecular kind of based approaches that we do have in the lab, PCR or variation real-time PCR. So literally being able to tell if a patient has a sample that's positive or negative in real time by monitoring the change in fluorescence for a target is something that we do use more and more, becoming more popular in the laboratory. An example of that is malaria PCR confirmation that we do offer at Alberta Precision Labs. The benefits are, there's sometimes significant cost savings associated. It's much faster than microscopy, as well as uh, serology, can be less labor intensive and very, increased, very high sensitivity with some of the targets that are available. And so PCR is kind of taking off in the world of parasitology, and we see, different variations of this. There are now commercial kits that are available looking at, say, for gastrointestinal parasites. You can now screen for a bunch of those different parasites causing gastrointestinal disease that uses a form of PCR, and it's just a lot more efficient that way. And so a lot of labs are now implementing these more molecular-based approaches. Now, I want to talk about sequencing because that's been taking off in the world of parasitology. And so sequencing allows for rapid, high throughput sequencing of the entire genome of some of these parasites on a single instrument in a laboratory. And the drawback with sequencing currently is that it's limited in the sense of it's being used more so in research settings, just because with parasitology, there's very limited databases that we can have access to compare the results to. So anytime you sequence, you want to make sure you have a good reference database available so that you can compare the results. And sometimes if those sequences for a particular organism aren't there, it may be, difficult to identify. And another huge complication with sequencing parasites is their genome size. Some of them are haploid, very large genomes compared to bacteria and viruses that can be sequenced routinely. There's a lot of work that needs to go into, finding that perfect mix of, of extracting the sample and removing any sort of inhibitors. So thinking about stool samples, for example, if you want to sequence a stool to look for a specific parasite, there's so much junk in there, so to speak, that you want to make sure you're able to kind of tease everything else out and have that parasite nucleic acid available for you to sequence. But there's really great potential with sequencing platforms. And in terms of in a research setting, it's used for strain typing. You can perform molecular surveillance, trying to see if there's an outbreak ongoing. Are the isolates of a certain parasite related? Thinking about malaria antimicrobial resistance, we can actually now look for markers of drug resistance and tell whether or not a patient perhaps has some sort of mutation in the drug resistance gene contributing to the reason why that their current drug regimen is not working. And kind of what made the headlines was a few months ago, about 6 months ago, sequencing was used to identify this novel parasite causing a infection in a woman from Australia. So this parasite caused a CNS infection referred to as human neural larva migrans caused by this worm found actually in carpet venom snakes in Australia called Opscaris robertsii, and then it's an ascarid. So sequencing was really cool and useful here because it was the first time this organism was identified in a human, and sequencing was a technology that was being used because it was really hard to tell microscopically, or even looking at the live worm, which was actually pulled out of this woman, unfortunately, whether or not it was a known parasite that we've seen before. So sequencing can be definitely useful. It's a great technology that we're definitely exploring in the lab, and definitely has its perks. Now, talking about proteomics, so analyzing the protein profile of an organism, and so that's really kind of more so used as evaluation of biomarkers for tissue, cell types, and other disease stages. And the biggest advancement of actually using proteomics as a means of identification comes with mass spectrometry platforms. So the MALDI-TOF, which I'll talk about in the next slide, it's heavily used in the world of bacteriology and mycology, now less so in virology and parasitology, but there is kind of a push or a interest in looking into whether or not MALDI could be used for some parasitological infections. So MALDI-TOF MS, it's a large acronym, stands for Matrix-Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. So essentially, it's a really fast inexpensive method to detect an organism, and detect an organism by analyzing its protein profile and comparing it to a database, and can really produce results in a couple of minutes once you have the isolate ready for it to be run on this instrument. And so what happens is proteins that are found in a sample are ionized into these charged molecules that accelerate through a kind of vacuum, and based on the analysis of their mass to charge ratio, an identification or protein profile is spit out. And I don't know if you can see it on the bottom right over there, but that's kind of like a fingerprint that we use to identify organisms in the lab. And so this slide just highlights in Canada and the US. So NRCP is kind of like our CDC equivalent for diagnosing parasites in Canada. Microscopy is really kind of the gold standard for diagnosing a lot of infections. So thinking about bloodborne infections like malaria in particular, microscopy is high yield. Across the board for all these different organisms that I won't belabor, you can see microscopy really still kind of the mainstay for diagnosing these bloodborne infections. And then thinking about intestinal parasites, so those that cause, gastrointestinal disease to some extent, serology and microscopy is still there. So as I talked about, those two methods are kind of, outdated. They are still useful, but really kind of the mainstay in these big labs that perform a lot of diagnostics for us. And so really, we have a need for implementing newer diagnostics to combat the declining expertise in, say, microscopy, for example, and improving turnaround times. And, sequencing and using proteomic platforms like MALDI-TOF are definitely potential ways to combat that. But how else? How can we actually assess for new diagnostics? What are the factors you have to consider when trying to think about what is the best diagnostic modality that can be used for a parasite? And so diagnosing a parasitic infection, which we'll talk about, is quite complicated, and it's essential to diagnose a parasitic infection for effective treatment and prevention of any complications. But there's a number of factors to consider. So the symptom onset, patient history, geographical location, epidemiological factors. So what do you think that parasitic infection the patient's presenting with has? But thinking about it from a lab perspective, what's really important to make sure to ensuring that the sample that you are collecting will be of the utmost value when you're trying to diagnose the infection. So performing, serial testing and getting the timing right, which we'll talk about, like parasite life cycles and how that plays into being able to capture that sample at the right time. The actual collection of the sample and handling of it, limitations with sensitivity and specificity depending on the test that you're thinking about. There's always issues of cross-reactivity with some of these helminthic infections, and having overall the expertise and equipment available. These are all kind of factors we consider on the lab side for when we want to think about having a diagnostic test. And so, thinking about the timing and serial testing, well, there are a number of factors that actually go into that. So thinking about the parasite life cycle, first of all, you can see on the bottom right, I've included a life cycle from the CDC of schistosomiasis, which is a parasite known to cause infection in Africa. So for example, this parasite has a very complex life cycle, many stages that are not necessarily present in the host body. The timing of your sample collection has to coincide with the presence of the right stage. For example, in the case of intestinal parasites, you want to make sure that the shedding of eggs and cysts and feces occur periodically. So you want to capture those samples at the right time to increase your yield of positivity. There's issues with some parasites. Some infections shed their eggs intermittently, so that meaning that the parasites aren't actually continuously present in clinical samples. So that's why you have to often collect multiple samples. So just because you get one negative result doesn't necessarily mean the patient, you can exclude that infection in a patient. There, you have to collect multiple samples. Clinical symptoms, again, is really telling for, when you can collect some of these samples. For example, in the case of malaria, parasites are detected in the blood during febrile phases of infection. And so you want to collect the blood sample when the patient is symptomatic, which can increase the yield for diagnosing it correctly. Diurnal variation, so certain parasitic infections, again, for example, Schistosoma mannsoni, so schistosomiasis, the release of eggs into the host bloodstream follows a diurnal pattern. So they peak, the egg output actually peaks in the morning. So you want to make sure you collect the samples at the appropriate time of day, similar with filaria. So there's a number of different, parasites that have all these different variations that you have to consider when you want to collect a sample, because that has implications for us being able to increase our ability to diagnose the infection or catch the infection in the lab. Some infections have transient parasitemia, for example, Trypanosoma brucei, causing African sleeping sickness, where parasites may appear and disappear in the blood. Again, another reason why you want to collect multiple samples over time. There's seasonal variations and treatment effects. All of this contributes to the complications that we face with trying to diagnose a parasitological infection. So anyone involved in this process, it takes a lot of solid understanding of the parasitic infection in question, making sure you're able to collect samples at the right time, that they're in the right, form, and making sure that, the lab that you're sending these samples to have the appropriate resources available to diagnose these infections. And so I kind of did talk about, sample collection and handling, and I don't want to belabor that too much, but a lot of that actually can factor into whether or not we are able to identify some of these infections. So I don't want to run out of time, but I'll briefly talk about leishmaniasis and, how even with this parasitic infection, diagnostics can be a bit complicated. So leishmaniasis is a protozoan parasite that is transmitted by a sandfly, and it's predominantly found in the tropics. So what happens is, the sandfly will take a blood meal, inject what is referred to as a permastogote into the human. These permastogotes are then, engulfed by macrophages, and depending on the actual species, they can remain localized or disseminate further into the bloodstream, resulting in different forms of infections. So within leishmaniasis, there's three kinds of disease presentations: cutaneous disease, so destruction of your skin. You can have mucosal disease, which is the extension of that into more your mucosal lining, mucosal membranes, or visceral leishmaniasis, where the parasite infects your internal organs, resulting in severe morbidity and mortality. And so a lot of these disease presentations are multifactorial in the sense that there's the infecting species. You have to think about the geography. Where is the patient from? The host immune response can play out into how the disease presents. There's virulence factors associated with the parasite that contribute to more severe disease, as well as the location of the sandfly bite. So all of that kind of plays into these different disease presentations you can have even within one parasitic infection. And so with leishmaniasis in particular, we group the different species into what's old world and new world, and there are about 20 different species, and there's more to come as we use our genomics and identify new species. But essentially 20 different species that can cause the spectrum of disease, and combine that with your host immune responses, you can have different presentations. Virulence factors are highly important to consider with regards to the disease presentation as well. So what inherent to the parasite can cause it to produce a more severe disease or a specific kind of form of the disease versus the other. And then leishmaniasis in particular, there's a number of key virulence factors that are kind of attributed to certain disease states. There's also species-specific genomic differences. So, those that come from the old world, if you have a patient coming from the old world, you're often thinking of certain species of the old world leishmaniasis group compared to the new world. There's differences in their genome that can contribute to why you have one form of the disease versus other. So all of that kind of, goes into how the parasite can cause infection and what that means for us being able to diagnose these infections in the lab. So with diagnostics for leishmaniasis, for example, microscopy, as I did mention, is still our mainstay. So we can look at these organisms under the microscope on a variety of different samples. And if you recall my slide on the life cycle, you can see what is referred to as the permastogotes. So on the bottom right, there are these flagellated parasites that are kind of swimming. Whereas, if you look for the amastogote, which is the internalized or the transformed stage of the parasite within the macrophage. You can see kind of these like kidney bean dots within the macrophage, and that's diagnostic of leishmaniasis. But being able to identify these guys can be tricky to the inexperienced technician. Now, culture is often used to grow these organisms, but it's very hit or miss. It takes a long time to grow these organisms in the lab, and so when you're thinking about an infection with leishmaniasis, microscopy is kind of like your top tier diagnostic test you want to use. Having culture done is also useful. And those are kind of the two main modalities that we use in the lab, as well as molecular testing. So using polymerase chain reaction, there are different variations looking at sequencing incorporated as well. So all of that is kind of very useful in diagnosing an infection. Now, what's interesting is serology is kind of limited to one form of the disease, the more severe form of the disease, visceral leishmaniasis, just because it relies on the presence of a certain immune response that is not present with the other two forms, cutaneous and mucosal disease. So even within, as I mentioned before, one disease state, one disease, you can have different, disease manifestations, and all the diagnostics can vary between them just because of how the parasite is genomically, as well as the host immune response. So a number of things to consider when you are diagnosing even just one infection, leishmaniasis. And so I don't want to take up too much time. I know it's 2:15. I just want to leave some time for some questions. I just want to kind of reinforce the fact that, parasitological infections are becoming increasingly encountered. I'm sure you've probably seen them in the news with malaria and a few cases in the States. We're encountering them more and more just because, we like to travel. There's societal social behaviors, vectors, and the organism itself. All of those geographical distributions are changing because of climate change and expanding with urbanization, et cetera. So understanding these parasitic life cycles are very important because that informs the diagnostic modalities that you would actually implement, and combining that with, the appropriate sample collection, timing of that, the sensitivity and specificity of each test that can exist, and the expertise available. All of that kind of complicates the ability to correctly diagnose an infection. And so I just wanted to give you guys kind of an appreciation of how diagnosing parasitological infections in the lab can be complicated. It's interesting. It's kind of like detective work for me. And it's always improving or always getting better. There's novel diagnostic assays that are coming into play. People are looking at how AI can actually help address some of the current gaps in parasitology. So using AI to read microscopy slides, and if they're negative, they just get reported as negative. But if they see something suspicious, maybe they'll flag that to a technician or a technologist for further review. So that's a way in which, AI is kind of being looked at and being incorporated into maybe the diagnostic algorithm for certain parasitological tests. So with that, I'm happy to answer any questions. I put my email here. If you have any questions at any other time, happy to answer them. And thank you so much for your time.